Good day, Dr. To and fellow classmates. Our group is going to discuss on the following journal paper titled A Simple and General Approach to Assay Protease Activity with Electrochemical Technique. Proteases are enzymes that cleave peptide bonds and are involved in a variety of diseases. For example, in HIV, there is a HIV protease. The authors want to develop a biosensor to assay protease activity because they feel that existing methods such as HPLC and ELISA are more suitable for quantitative analysis. One approach involves fluorescent labeled peptides to probe protease activity. However, the use and preparation of such modified substrate tends to be complex and costly. Thus, in this paper, the authors aim to develop a biosensor using unmodified substrate. They use trypsin as the target model. How does this biosensor work? Firstly, the peptide is immobilized onto the surface of the electrode. This peptide carries a positive charge, after which the electrode is immersed in a sample solution. In this case, trypsin is not present. Differential pulse voltammetry is being run using a positive redox species. In this case, it's a positive ruthenium complex. As both the peptide and the redox species carry positive charge, there is an electrostatic repulsion between them, thus blocking the redox species from interacting with the electrode. As a result, a very weak signal is detected. However, if trypsin is present, it will recognize the specific site and cleave the peptide, leaving a neutral fragment behind. This reduces the positive charges on the peptide, making the blocking effect against the redox species weaker. Thus, more redox species are now able to interact with the electrode to produce a strong signal. In order to prove that this is indeed the mechanism, the authors tested the biosensor using a negative redox species in the absence of trypsin. As shown in the graph, even in the absence of trypsin, a pair of strong redox waves could still be observed, suggesting that the peptide surface can only block out positive charges. However, our group feels that the authors should have also tested the negative species in the presence of trypsin. This is to ensure that the signal measured when trypsin is absent is similar to that of when trypsin is present. With that, this pair of signals can be compared with those obtained when positive species were used. This can then further confirm that the peptide and the positive species are suitable for assay of trypsin. The authors have also tested a range of concentration of trypsin using the biosensor. From graph A, the signal increases as the concentration of trypsin increases. This is so as trypsin decreases the number of positive charges, which allows more redox species to interact with the electrode, hence producing a stronger signal. A working linear range and detection limit are obtained from the calibration curve of current against concentration as shown in graph B. This table shows the comparison of the biosensor developed by the authors against other similar researchers that are based on unmodified substrate. We can see that the author's biosensor displays a lower detection limit than most of the other methods. In order to ensure the specificity of the biosensor, the authors also tested it using bovine serum albumin, or known as BSA, and human hemoglobin. It was found that low signals were detected when they are used instead. These differences suggested that this biosensor could be specific for detection of trypsin. In addition, the authors also added these proteins in the presence of trypsin and found that there was little interference. We can see from the table, the signal ratio of trypsin alone against trypsin in the presence of interfering proteins is about 1:1. However, our group feels that the authors should have used other proteins such as serine protease on top of BSA and hemoglobin to ensure the biosensor specificity. This is because BSA and hemoglobin have no enzymatic reaction on the peptide. Furthermore, there might be other proteases that can cleave the peptide, producing a false positive result. In order to prove that the biosensor can work in complex matrices like serum, they spiked the serum sample with trypsin and found that the signal of the biosensor in serum and buffer are similar. But our group feels that the authors should have used a control like a serum sample without trypsin. This is to ensure that the signal detected is not due to other compounds in the serum. Additionally, the authors failed to compare their method against existing methods like HPLC or ELISA to detect the concentration of trypsin. We feel that the comparison should have been done to ensure that the result produced by the biosensor is reliable. The authors also carried out an additional experiment to prove its versatility. By changing the peptide, the biosensor can detect other proteases. From the graph, we can see that by changing the peptide, it can detect other proteases like thrombin. Despite the lack in certain aspects, we feel that the authors have carried out a rigorous experiment to demonstrate the reliability of this biosensor. With that, we have come to the end of our discussion. Thank you for watching our video.